Welcome to section 10.3. Now in 10.2 we finished off talking about independent assortment and how what you get for one gene, whichever alleles that you happen to get, that does not impact what you get for a different gene. So essentially you can treat each gene as its own separate box that you can reach inside and pick out what you're going to be. But what you do for the first box doesn't impact what you get for the second. Now we're going to talk about something called linkage. Now linkage is going to be the opposite of this. So linkage is going to be where we now know that genes are located on chromosomes. So because we know these genes are on chromosomes, the trick is there's many genes on each chromosome. So as long as genes are on separate chromosomes, they'll be independent assortment. So independent assortment, this idea, is going to have to be where the genes are on different chromosomes. And that's usually the case, but not always. There are plenty of cases, although not really most of the ones that Mendel studied. So most traits are on separate chromosomes, but some of them are going to be on the same chromosome. And so that means it's now a package deal. So you do tend to get those things together. And so this explains how sometimes we will see traits that are usually packaged, such as pale skin and red hair. If these genes are on the same chromosome, and if I get that chromosome when independent assortment happens in meiosis, I now get both of those alleles that they possess. And so that might be, in the case of our peas that we used, it might be tall and purple flowers. Now those aren't actually linked, but if they were, we would tend to see tall and purple flowered offspring or short and white flowered offspring, because we can't mix and match. You just got to go with what you get pre-packaged on a chromosome. Now, there are some instances where you will get recombination, where you'll get mix-ups. So even though in the case of tall and purple, if we assume that they're somehow linked, then we'd expect to see tall and purple offspring or short and white. They'd be always packed up, they'd always look like one of the parents. There are still some cases that this doesn't hold true, and that's because of our good friend crossing over. And crossing over can break linkage because it can allow these two chromosomes, one of which codes for tall and purple, one codes for short and white, they can swap pieces. And so based upon where exactly those genes are located on the chromosome, especially if they're farther apart, there's a decent chance that you're going to get some of your chromosomes during crossing over, during meiosis, that will swap, and so they will break linkage. They'll make it now where this one might have tall and white, and this one has short and purple. Now, this only happens a smaller degree, so you'll still tend to see where most of the offspring look like a parent, but you will see some of them that don't. So for instance, you might get where when you're analyzing these guys, you might get that 40% are tall and purple, 40% might be short and white, so if we kind of draw these, and then you might have 10% are going to be short and purple, and you might get 10% that are tall and white. And so it's a smaller amount. You can definitely tell there's linkage here because it's not 25, 25, 25, 25, which is what we expect. You know, when we're doing this type of a cross, we're typically going to be crossing someone that is heterozygous for both with someone who's homozygous recessive for both. And we're going to do a lot of practice with Punnett squares, so this will make sense to you after you're set in class, but it's just going to take a lot of time if I ran through a ton of these outside of that. I might do a Punnett square video separate just to make sure you know how to do this. But if you do this cross, we expect to see 1 to 1 to 1 to 1. And so that would equate to having 25% of each possible phenotype combination. When you don't see that, that implies that there's linkage. That implies that these two genes are on the same chromosome, and that explains what we're seeing. Now, the other cool thing that we can see that goes on that has to do with chromosomes, that's kind of us going a little bit past what Mendel did. We're going to start moving into, over the next several chapters, some modifications to what Mendel did, because he just laid the groundwork. And that was amazing what he did with what he had. But we found a lot more complicated scenarios since then, so we've added to it quite a bit. Now, one of these things that we can see that happens one of these things we can see that genetically happens with reproduction is a kind of screw up, and especially in plants this is common, and we call this polyploidy. So we've talked about how right now we are diploid, so our cells are typically 2N. We have two sets of chromosomes. We've talked about how when we go through meiosis we produce 4 gametes that each are 1N, and that's essentially what we're going to pass on to our offspring, and the other parent does the same. We can quick draw that out just to make sure we're not confused. So this is normal, all right? Now, sometimes though there's a screw up where you don't properly split the sister chromatids during meiosis II. And so what happens is you get some gametes that have 2 sets because they grab both sets, which means another gamete has none. Now that gamete's just going to be not usable. In most cases, you're just not going to make it. But the one that's 2N could still pair up with another one of these ones that's 1N, and you could get an offspring then that has 3 sets. We would call that triploid instead of diploid. 3 full sets of chromosome. So with polyploidy, in plants we'll see they oftentimes do better if they're polyploid. So we've got some things like strawberries that can be in the neighborhood of 10 or even 12N. Animals, not so good with this. Typically, if you're an animal and you're polyploid, that usually results in death. There might be one exception, at least, that we're aware of, one type of rat that I haven't fully checked on. But in general, with animals, this is very bad if you become polyploid. But for plants, oftentimes you're better than you were when you weren't polyploid. And oftentimes in plants, you can get where polyploid plants can go through these events with other polyploid plants. So, you know, a 3N plant can screw up, and then it goes with another plant, and so now it becomes 4N, and then that plant screws up at some point, and you're 6N, and this can just keep going. There's a limit at which you just have too many chromosomes, and I'm sure it falls apart. But we do see some plants that have upward of about 1,000 chromosomes. So plants, when it comes to chromosomes, are crazy. But it allows for us to get a lot of the crops that we have that produce high yields, because a lot of the crops we use are 6N, 10N, you know, they are polyploids like this. So at the same time, you should be thankful that these polyploids exist because it means more food for us.